And then in the cell, there are enzymes that can build up that GTP, GDP into GTP again. And GTP is never regulated. And now I'm in the on form, so it's the guanine nucleotide that's changing it on. So I'm causing some reaction to go, 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 until the gap causes me to hydrolyze that again. Question?
It's a new GDP?
Pardon?
The GTPase, did you say it's a new one?
Yes, so the guanine nucleotide exchange, so these GDP, I'm holding GDP, and then in the cell, it's never is lots of GTP, and the guanine nucleotide exchange factor takes one of those GTP and will exchange it on the GTP, so it's now in the on form. Does that answer your question? So it's a new GTP molecule. These will all get built up into new GTPs. They might be used for this GTPase, they might be used for another GTPase, they might be used in DNA synthesis or something like that. So all the things that guanine nucleotides are used for in the cell.
So it's going from on to on in the cycle, but the enzyme itself is not specifically being changed because it's adding in a new one.
I think the answer to your question is yes.
It's being replaced.
Yeah, it's being replaced, that's right. And again, there's a small conformational change between the GTP bound form. That's how this works as a regulatory enzyme. The GDP is a different shape than the GTP form. And when it's in its GTP form, it'll interact with some cellular reaction to cause that to go. When it's stimulated, when it splits its GTPase, then it's in the off form, and its shape changes so it can no longer stimulate whatever reaction this GTPase is responsible for. There are probably 50 different GTPases in each of your cells, different ones, and they're all responsible for a different reaction of some kind of cell biological reaction. We'll be covering some 5 of those or so in here, but the principles are always the same. The GTPase is on in its GTP bound form. It's off in its GDP bound form. There's a gap that stimulates that side, and there's a GEF that stimulates that side. Other questions? Arturo?
Maybe just to
clarify something?
Give an example of a protein that you already know. We didn't talk about GTPase involved in that, but the SRP, the protein that recognizes the ERSS, is a GTPase, and it binds and unbinds by cycles related to GTPase. The SRP receptor is also, and their interaction is turned on and off by cycles of GTPase. And you don't need to know the details of that, but that's one example of things that you already know. 2.
Yeah. Other questions? Okay, this one you need to know. We're going to talk now that we understand how small GTPases work, we're going to talk about how they work in membrane trafficking. Because we're going to talk about a small GTPase called, SAR1. So that's the name of a particular small GTPase. It was discovered at a yeast genetic screen, so it has a yeasty name, which is meaningless. The P means the protein instead of the gene. So the SAR1 gene, the SAR1 protein, SAR1P, is a small GTPase that undergoes this cycle. And what it controls, the reaction it controls, is the assembly of coat proteins on the ER. So we have talked about clathrin coats and how they form at the plasma membrane and at the trans-Golgi network. And now, so clathrin coats. Plus, so clathrin plus AP2 is responsible for traffic from the plasma membrane to endosomes. Can you everybody see this right here on all parts of the room? Can you guys see this okay? Yeah, clathrin plus AP1 goes from the trans-Golgi network to pre-lysosomes. So that's the coat protein, that's where it goes. The receptors, we've covered one of those a little bit. The LDL receptor is here. We'll later cover this receptor. And now we're talking about COP2 coats. And these are responsible for going from the ER to the cis-Golgi, the first step of the Golgi apparatus. So what's going to go in these vesicles from the ER to the cis-Golgi? It's going to be everything that's along the secretory pathway. So all those proteins that began with an ERSS that are going to end up on the outside are going to initially be in COP2 coated vesicles, and that's how they're going to leave the ER and go to the cis-Golgi. So there's going to be all sorts of proteins here, receptors, that are going to be secretory proteins. And now I said that there was a small G protein that regulates the steps. And what this small G protein is responsible for, if you think about all these steps of going, I'll tell you right now that COP1 coats go from the cis-Golgi back to the ER. And we'll fill in this table as we talk about these steps. So let's consider the cytosol. It's full of these proteins. It's full of clathrin, and it's full of AP2. It's full of more clathrin and AP1. It's full of COP2-coated proteins. It's full of COP1-coated proteins. We saw the movie of the triskelions assembling on the plasma membrane, and those same principles are used by COP2 proteins. They're individual monomers soluble in the cytosol, but they're only going to assemble on the endoplasmic reticulum to bring proteins to the cis-Golgi. Whereas in the clathrin's going to ignore that. The COP1 coats are only going to assemble on the cis-Golgi and take proteins to the ER. So how do these proteins recognize and go to the appropriate membrane? How is this step regulated, coat formation? And that's what these small G proteins are going to regulate. And we're going to focus, because the story is most understood for COP2 coats, and we're going to focus, the small G protein there is SAR1. SAR1P for the protein form. So let me show you how that works. So SAR1P is a small GTPase, so we know that it is on in its GTP-bound form and off in its GDP-bound form. And the reaction that SAR1 is coupled to is the assembly of COP2 coats on the ER. So let's see how that works. So that's what it's right here in this diagram from your book right here. This shows the ER membrane right here, and we can see COP2 subunits. So they're sort of there are many isoforms of COP2. We're going to call them all COP2. This is not a case like clathrin was, where there were two layers, AP1 and AP2. All the COP2 coats sort of come on at once. And we see that those are forming on the ER membrane and pinching off a vesicle. And in that vesicle is going to be any receptors that recognize the COP2 coats, as well as anything that they're bonding. And as we saw before, the coat is sort of transient, it's going to fall off. So the way that SAR1 regulates this step is when it's in its GTP-bound form, active, SAR1 promotes coat assembly. So as long as SAR1 is in its GT-bound form, it's going to regulate coat the assembly of COP2 coat proteins, not clathrin or COP1, but COP2 coats, and that will cause the budding of coated vesicles. Once SAR1 is in its GTDB-bound form, it's inactive, so the coats will no longer assemble. So you can think of it as promoting coat disassembly and the formation of uncoated vesicles. So how then, so we see that SAR1, so that's this little yellow protein right here, when it's in its GTP-bound form at the ER, so as long as it's there, it's going to promote coat assembly and cause the formation of these. So SAR1 regulates coat assembly and disassembly right here. So how do we get that then assembly to occur on the ER and not on any other step? How does this regulate coat assembly only at the endoplasmic reticulum? And this is pretty cool, I think. It turns out that the SAR1 gap, so this protein right here, that's going to exchange the GDP for, so take SAR1 that's inactive and exchange it for GTP. That's an integral membrane protein that is localized on the endoplasmic reticulum. And that's what we see right here. So here's the SAR1 gap. And it also turns out SAR1 is a little bit special in that it has in the protein sequence an amphipathic helix, meaning it has a little hydrophobic tail, a hydrophobic helix of amino acids. And SAR1, when it's in its GD, these have different conformations in the GTP and the GDB bound form. And this amphipathic helix right here is hidden when SAR1 is in its GTP bound form, but it sticks out once, that's the amphipathic helix, it sticks out once it's in its GTP bound form. So if we place the SAR1 gap right here on the ER membrane, and then we have SAR1 when it's in its GDB bound form, this amphipathic helix is hidden, and so it's a soluble protein floating around in the cytoplasm, along with all the other co-proteins and everything like that. So it's just floating around, making its own business. And then when it hits the SAR1 gap, so I'm floating around with the membrane, and then I hit the ER membrane, and I encounter the SAR1 gap, and that will exchange SAR1 into its GTP-bound form, and then it'll start promoting its reaction. And its reaction is to promote the assembly of COP2-coated vesicles on this membrane. So as long as SAR1 is in its GTP-bound form, coated vesicles, these coats will form, and they'll bud off. And it'll keep going on. So once SAR1 is, so once in this vesicle then, it turns, what makes the SAR1 turn off? What happens is it turns out that it's a weak gap weak GTPase activity, weak gap activity, that's the word I'm looking for, weak gap activity. These COP2 coats themselves, coat proteins, have weak gap activity, but when there's so many of them, and there's a conformational thing when they're in a round vesicle, they are the gap that will cause the SAR1 protein to hydrolyze GTP into GDP. So once that happens, you can see that little amphipathic tail right there. It'll go, the gap will cause it to flip its tail up, and so this coat will fall off, and it will be a naked vesicle containing the receptors, because those are stuck in the membrane, and whatever the receptors are bound to, but there's no coat there. So the COP2 vesicle then is ready to fuse with the next compartment with the cis-coating network. Does that make sense? Okay, so where's the confusion?